shall now move on to our next speaker, Dr. Kee Boon Pin, senior lecturer from the Department of Biomedical Science as well. So let me just introduce Dr. Kee briefly. Dr. Kee has graduated with a doctorate degree from the University of Malaya as well in 2014. His research interests lie within molecular genetics with a recent focus on the genetic investigation of cardiomyopathy among Malaysian patients via next generation sequencing and bioinformatic platforms. Besides, his expertise also lies in population genetics and mitochondrial DNA. Dr. Kee carries out genetic association studies to investigate the relationship between genetic markers and the onset of disease in Crohn's disease and systemic lupus erythematosus patients. Dr. Kee also works on the identification and differentiation of microorganisms using various molecular techniques. He has experience examining and analyzing genomic data generated from markers in different DNA systems, including mitochondrial DNA, autosomal, and sex chromosomes. Dr. Kee has won several awards in the Research, Invention, and Innovation Expos conducted at UM in the past. It is my pleasure to invite Dr. Kee to present his talk entitled Unlocking Heart's Genetic Insights into Cardiomyopathy in Malaysia. So over to you, Dr. Kee.
Yeah, thank you, Dr. Bavani. So a very good morning to everyone who is watching this seminar. First of all, thank you to Visibility Unit of the Faculty of Medicine for hosting the breakfast at UMHealth, and also thanks to Dr. Bavani for moderating these sessions. So I'm Kee from the Department of Biomedical Science. I'm happy to have the opportunity today to share with you one of my research directions about the genetic studies that we conducted to understand the genetic etiology of cardiomyopathy in Malaysia. So first thing first, what is cardiomyopathy? Cardiomyopathy is a disease of the heart muscle. It's also collectively known as a disease that affects the heart muscles and make it harder for the heart to pump blood to the rest of the body parts, which can then ultimately lead to various symptoms. The initial symptoms of cardiomyopathy can be mild and common as other diseases, such as shortness of breath, fatigue, swelling of legs, chest pain, dizziness, fainting, and coughing. However, if it's left untreated, these symptoms can progress to a more severe stage, which then ultimately results in heart failure. The treatment for cardiomyopathy includes medication and sometimes surgically implanted devices and heart surgery, depending on the severity of the patient's conditions. So in Malaysia, cardiomyopathy was reported to be responsible for 28% of heart failure cases. Cardiomyopathy comprises two main subtypes that are dilated cardiomyopathy, DCM, and hypertrophic cardiomyopathy, HCM. DCM is the condition whereby the ventricle is enlarged due to abnormal contraction, which results in weakened pumping force for the heart. DCM is usually diagnosed with echocardiography with ejection fraction less than 45% or fractional shortening that is less than 25%. Sometimes, cardiac MRI can also be done to observe detailed images of the heart structure. The prevalence of DCM is about 1 in 2,500 individuals, and it was reported to have higher prevalence in African, followed by Caucasian and Asian. Male tend to get DCM more frequently than female. On the other hand, hypertrophic cardiomyopathy, HCM, is a condition whereby the ventricle septum is thickened, making the heart stiff to pumping blood. The diagnosis of HCM is also based on echocardiography, with left ventricular wall thickness more than 15 mm. Similar to DCM, sometimes cardiac MRI and biomarker testing can be done as well. The overall prevalence of HCM is 1 in 500 individuals, and is more frequently occur in African than Caucasian. It also is also more predominant in male than female, with a ratio of 2 to 1. Okay, so cardiomyopathy can be caused by various factors, such as infection, toxicity, or other complications like metabolic disorder or neuromuscular diseases. However, CMP is also known to have a strong genetic predisposition, whereby 25 to 50% of DCM and 30 to 60% of HCM cases are found to be associated with genetic factors. So the presence of multiple genetic variants is usually linked to more severe conditions of cardiomyopathy. So the genetic causes of cardiomyopathy has been extensively studied worldwide in various populations. Today, more than 100 genes have been implicated with CMP. These genes are usually involved in different functions and structures of the heart, such as the cytoskeletal proteins, calcium ion channels, sarcomere structure, and nuclear envelope. Among them, MYH7, MYBPC3, TNNT1, TPM1, and LMNA are genes that have been reported in both DCM and HCM. So as I mentioned in the previous slide, CMP has been extensively studied in many populations at different scales, especially in the Western population. However, there's limited data on the genetics of CMP in the Asian population, especially in Malaysia. The genetic etiology of CMP in the Malaysian population remains unclear, which warrant comprehensive study via advanced molecular and bioinformatic approaches. Therefore, our team decided to initiate the genetic study on cardiomyopathy by working together with our collaborators in UMMC and IJN. Our team consisted of researchers with molecular expertise and clinical cardiologists from both UMMC and IJN. We also recruited CMP patients in both centers. Our CMP cohort consists of more DCM cases than HCM, and the patients are predominantly male. In terms of ethnicity, we have more Malay DCM patients and Chinese HCM patients. So in the past few years, we have carried out several genetic studies in the CMP cohort. So in this presentation, I would like to share with you 3 of our completed studies and what we have learned in these studies. So our CMP genetic study started with the sarcomeric point mutation study. In our first study, we examined a total of 20 most frequently reported mutations in a Caucasian-based CMP cohort. So this mutation accounted for 11.8% of the total mutation observed among this Caucasian cohort. Therefore, we are thinking that this mutation might also be present in the Malaysian cohort. So among the 20 mutations, 13 were pathogenic, 3 were lightly pathogenic, and 4 were variant of uncertain significance. The genotyping of this marker was then carried out by T-ARMS PCR genotyping assay in 152 CMP samples. So our findings show that out of all the mutations that we genotyped, only 1 heterozygous mutation carrier was found among the 152 Malaysian CMP patients. This constituted 0.7% of the CMP cohort or 2% of the HCM cohort. The other 19 mutations were all absent in our cohort. So the notes taken from our first study is that the prevalent markers in other populations may not as prevalent in our population due to the genetic background of different ethnics and population. So in our second study, we decided to perform full gene sequencing for CMP-associated genes, especially in the genes where the mutations were absent in our first study. Therefore, we amplified the full sequence of 5 genes, including LMNA, MYH7, MYBPC3, TPM1, and TNNT2. So sequencing was performed by a next-generation sequencing platform, and in this study, we hope to discover mutations that are associated to CMP development in the Malaysian cohort. Out of the 105 sequenced CMP samples, we managed to identify 28 variants among 31 CMP patients, which is about 30% of the cohort. So among the discovered variants, 5 were pathogenic, 8 were lightly pathogenic, and 15 were variant with uncertain significance. We also identified 4 novel variants, which have not been reported in databases like DBSNP and EXACT. Our finding is consistent with other studies, whereby MYBPC3 and MYH7 remain as the most dominant genes for the Malaysian CMP cohort. Hence, in the second study, we realized that the prominent CMP-associated gene will still remain relevant, although it's in different populations. In our third study, we wanted to sequence the whole mitochondrial genome of the CMP samples. This is because heart is a heavy energy-dependent organ. Therefore, the intact function of mitochondria is important to maintain constant supply of energy to the heart. Defect in mitochondrial function due to genetic variation in the mitochondrial genome would essentially resulting in inadequate energy supply to the heart and causing cardiomyopathy and heart failure. We amplified the whole mitochondrial genome of the 146 CMP patients and subjected to sequencing by next-generation sequencing platform. Through the whole mitochondrial genome sequencing, we discovered 20 pathogenic, 26 lightly pathogenic, and 171 variants of uncertain significance. We also identified variants with confirmed pathogenicity such as G11778A and A1555G in our CMP cohort. Some of the variants also present in both DCM and HCM cohorts, such as G137088A and T12811C. A total of 27% of DCM patients and 26.7% of HCM patients carried the pathogenic variants in our study. So this highlights the fact that other than the autosomal genome, mitochondrial variants may also contribute to the development of CMP as well. So based on the findings in our studies, we suggest that it's crucial for more comprehensive genetic studies conducted on the CMP patients to discover the full genetic landscape of CMP etiology for the Malaysian population. So with this genetic data and together with other data, we can improve the diagnosis and management of CMP patients in Malaysia. So before I end my presentation, I would like to share some of our current progress and future plans for CMP research. Currently, our team is working on genetic association study of CMP patients. We hope to identify some genetic factors that are associated with CMP development. We also started on whole exome sequencing and whole genome sequencing for some of our CMP samples, hoping to discover more variants and genes that may be specific in the Malaysian cohort. Thirdly, we are now validating some of the pathogenic variants discovered in our previous studies via in vitro assay. And last year, we had started working on the gut microbiota study among the CMP patients, in which we hope that we can find some markers based on the gut microbiome, which is related to CMP progression. For future plans, we hope to engage in multi-omics study for cardiomyopathy and able to identify and develop rapid tests for clinical application. So I would like to take this opportunity to thank all the volunteer participants in our study, and also thanks to the various funders who provided funding for our CMP research. Last but not least, I would like to thank my amazing team. So without their dedication and commitment, this research would not be possible. That's all for my presentation. Thank you.
Thank you very much, Dr. Kee, for sharing recent updates among genetic variations among Malaysian patients for cardiomyopathy. So we have one question from our attendees, one of our attendees. So from for you, okay. Were those patients associated with mitochondrial dysfunction on Q10 as a self-medication or prescribed by their care providers? If self-medication is prevalent, it may have decreased the population afforded via sampling. This is from YK Chan.
Were those patients associated with mitochondrial function on Q10 as a self-medication or prescribed by their care providers? So the patients we sequence for the whole mitochondrial sequencing. So basically, there are also CMP patient diagnosed in UMMC and IJN. And then they are also on medication for cardiomyopathy. Yeah.
Okay. So I think there's a follow-up question, right? Was that like answered together? If self-medication is prevalent, it may have decreased the population afforded to your
self-medication. Self-medication in this case referring to their own medication other than the doctor's prescription.
I assume, yeah.
We decrease the population afforded to your sampling. I'm not sure about this question.
Perhaps I think YK Chan could clarify this, and that will be answered in the chat box later.
Yeah, of course.
I think he has a question. Can I present the question orally?
Yeah, of course.
Okay, I don't know whether he has access to unmute. Maybe admin can help. Or if we were due to time limitations, perhaps YK Chan can post your question in the Q&A box, and Dr. Kee can actually clarify that because the admin is not responding about unmuting yourself. So thank you for your question. So perhaps you can clarify the question in the chat box, and we will get Dr. Kee to address your question.
I will in touch with
Dr. Kee will be in touch with you. So maybe I have just one question about so when you say that there are mitochondrial variations versus autosomal genetic variation. So
Mhm.
is that possible that either one of these can be a superior reason for why, as a superior reason contributing towards cardiomyopathy, which or they can occur together, or one can actually dominate in
Okay.
resulting in cardiomyopathy in certain patients.
Yeah. Okay. Usually, it can both variation in the autosomal genome and mitochondrial genome can occur together. And then when there's accumulation of more pathogenic variant, then the condition of the patients tend to be more severe and then progressing faster. So
Mhm.
that also the impact of the variants also depends on the type of variants. Like if it's a frame shift, then it will actually cause much more severe impact compared to a nonsense or missense mutation. So that applied to both the autosomal and mitochondrial genome. So it depends on the type. Then it could be that one variant, when they occur together, it could be one variant is superior than the other, or
Mhm.
it could be both, yeah, equally impactful to the patient's condition.
Noted, noted. Thank you for the clarifications. So with that, thank you very much, Dr. Kee, for your sharing session.
Thank you,
And
Dr. Wani.
with that, I shall now pass the session to Professor Dr. Pung Shin Yi to moderate the last session for today's webinar. So I'd like to call upon Dr. Prof. Fong.